Assalamu alaikum and welcome back to another video. In today's video, we are going to discuss about structure of nucleosome. We are going to discuss about the packaging of DNA into nucleosomes. Now, first and foremost, what is a nucleosome? So the structure which you can see over here on your screen, that is a nucleosome. A nucleosome that's formed when the DNA, it basically wraps around the core histone proteins. So you can see from the figure here. So this is a histone core protein made up of different histone proteins, all of those positively charged proteins which are rich in lysine and arginines, those are the histone proteins. And now they form a core complex over which the DNA is wrapped around, and it makes around 1.65 turns, and that forms a structure called as nucleosome. So it gives a kind of a beads on a string kind of appearance. Now, why this structure is so much important? So let's first discuss what are the different proteins in present within this histone core. Now, if you see the eukaryotic cells, they commonly contain five abundant histones. Starting from H1, that is a linker. If you see this is a linker DNA, usually this is 20 to 60 base pairs. So this is H1 and rest, the core histone core, it's made up of H2A, H2B, H3 and H4. These are the core histones. And now second thing is that each of these core proteins, they are present as the two copies. Okay, so we have two copies of H2A, two copies of H2B, likewise two of H3 and two of H4. So this makes it a histone octamer. That's why we say it's an histone octamer. Now let's proceed ahead and see the differences. If we can see some of the differences between the different histone proteins. Now again the same, we have H2A, H2B, 3 and 4. Now from looking at their structure, so this is the N-terminus tail and this is the C-terminus tail. And this structure in the middle, it is a histone fold domain. Now, why is it important to distinguish them on the basis of their N-terminus tails or C-terminus tails? Although C-terminus tail, they don't hold much importance in terms of certain functionalities. So our prime concern should be N-terminus tail. But here you can see that the C-terminus tail in case of H3 and H4, these are quite short as compared to the other two here. Now, these N-terminus tail, they hold much more significance. It is because the N-terminus tail is the region where the actual post-translational modifications they do take place. Whether it be acetylation, whether it be phosphorylation, whether it be methylation, all of those modifications they happen over the N-terminus region. So we saw the structure of the histone core and the nucleus which was wrapped around it. And but the structure which we saw, in that wrapping up of the DNA, we will not see the involvement of these N-terminus. So these N-terminus, they will be always, so the histone core is made up of the histone proteins, but we'll see the tails, they will be lying outside of this core. So they don't play a basic function in the wrapping up of the DNA. Okay, so the most important role they play is the post-translational modification, and eventually that will help in making the DNA more accessible for the other processes like be it replication, be it transcription. So basically making it more active, or certain other modifications which will help in the condensation of the DNA. So that is the role histone plays over here. Now let's look at the structural aspects. We talked about the H2 and H2B dimer. We talked about H3 and H4 tetramer. Okay, so what happens? Now first and foremost comes the concept of assembly of nucleosomes. Now you see here, again, this is the DNA which is wrapped around, and these are the structures which we said earlier. So we'll see here, we have H2A, H2B dimer, and an H3, H4 tetramer. You can see here, over here, and all of those histone proteins, they are present with their N-terminus tail, which are present outside of the core. So that's what I was saying earlier. In the previous slide, we discussed all of the core proteins of the histones, they play a role in the packaging. But the N-terminus tail, they are more involved in the post-translational modifications and not involved in the packaging within the nucleosomal core. Okay, so that was the concept about it. Now proceeding ahead, we'll talk about the assembly of the nucleosome. So this assembly of nucleosome, that's initiated by the formation of first and foremost a tetramer, which we saw earlier. First and foremost, this tetramer, it will come and it will bind here. preceded by the binding to the double-stranded DNA. Now, after the tetramer is bound, the DNA it will recruit two further copies of H2A and H2B. So first a tetramer of H3 and H4, then a dimer of H2A and H2B come together, and all of this once assembled they form a complete nucleosome. So first it's the tetramer H3H4 and then it's the dimer H2A H2B, and then they wrap around the DNA and form the complete nucleosome. Okay, let's have a look at this structure here. So we talked about the histone core, and histone core it constitutes four different kind of proteins, and each of these proteins are present in the two copies. So we will see in the histone core two copies each of H4, H3, H2A and H2B they are present. And you can see from this structure the N-terminus tails they are protruding from the core of the nucleosome. Now, if this structure is given a protease treatment, all of these N-terminus tails they will be chopped off, and we are left with a structure here. So this structure where we have the core histones and the DNA which is wrapped around it, it is approximately around 147 base pairs. And this structure is the nucleosomes, which we can get after a protease treatment. So in this video, we talked briefly about the histones. We talked about the packaging of the DNA around the core histone, around the histone octamer. In the next video, we will see that how this packaging of the DNA on these histones, it helps in the chromatin assembly, or it helps in the chromatin architecture in the cells.